Welcome everyone. I am Mukherjee, and in this video abstract I will discuss our findings regarding how cancer cells sense suspended fibers. Cells of the tumor boundary continuously probe the surrounding fibers of various diameters by extending lateral protrusions. Over time, as the protrusions grow in length, the cells are able to detach from the primary tumor and begin invading into the surrounding stroma. In this study, we wanted to investigate how cancer cells sense suspended fibers of different diameters. In order to do this, we used the previously reported non-electrospinning step method to deposit a network of suspended large diameter base fibers orthogonal to suspended smaller diameter protrusive fibers. This mismatch in fiber diameters causes cell body migration to be constrained along the base fiber, thus allowing us to study the dynamics of individual protrusions in a repeatable manner. Thus, our reductionist approach can be visualized to recapitulate the protrusions extended by cells at the tumor boundary. Using our platform, we find that cells interact with suspended fibers by coiling, that is, they wrap around the fibers at the tip of the protrusions, as shown in this video. To investigate if the diameter of the fiber influences the coil size, we used the step method to vary the protrusive fiber diameter from the nanoscale to the micron scale, recapitulating the range of fiber diameters observed in vivo in a tumor microenvironment. It should be noted here that the diameter and curvature have an inverse relationship. Increasing fiber diameters implies decreasing the fiber curvature. Our findings show that the width of the coil is indeed modulated by the fiber diameter and increases with increasing diameter. We also questioned whether coiling was limited to only metastatic cancer cells. To determine this, we investigated coiling dynamics of non-tumorigenic MCF10A and compared it to the metastatic MDA MB231. We found that compared to the MDA cells, the MCF cells exhibited significantly diminished coiling behavior. In fact, this trend of larger coil width for the more metastatic MDA cells was maintained across the entire range of fiber diameters tested. Thus, in conclusion, we find that cancer cells biophysically interact with the ECM mimicking suspended fibers by coiling at the tip of the protrusion. Furthermore, the coiling dynamics is modulated by the curvature of the fibers. Finally, we find that in comparison to non-tumorigenic breast cells, metastatic breast cancer cells show significantly larger coiling widths. Moving forward, we would like to further investigate the key intracellular players that regulate coiling. Additionally, we would like to investigate the effect of adding a chemical gradient on coiling dynamics. And finally, we would like to identify the potential utility of coiling in the metastatic process. For example, if we were to visualize the coiling behavior as a continuous drilling process, could it contribute to invasion in other tissues such as the bone? We would like to extend a special thanks to Ellen Boggs and James Archwell for help in production of this video abstract. We would also like to thank the Institute for Critical Technology and Applied Science, the Macromolecules Innovation Institute, and the Department of Mechanical Engineering at Virginia Tech. Finally, we would like to thank the National Science Foundation. We thank you for your time and your attention.